This is the review video for um, Lecture 7, Immune Systems, for Unit 2. So this is the review video for the Essentials of Adaptive Immunity. So there are a few parts of this, this lecture, and you kind of need to hammer all of them when you study. So the study guide for this part isn't very long, but you need to know it all pretty well, because this is the foundation for learning the adaptive immune system in Unit 3, and without that foundation, it's going to be impossible. So let's get into it. The first topic is antibodies. So let's find antibodies in the slides. Let's keep going. So there's a whole section in the slides on antibodies. And what you should know about antibodies includes their structure. What are they made of? They are proteins. And so if you were to draw a three-dimensional picture of them, it would be a giant blob, but it would have recognizable shape because it has two heavy chains. Those are two long proteins that are covalently bound to each other, and then two light chains. Those are shorter proteins. So that's its overall structure, but it also has an antigen binding site, two of them, or depending on what kind of antibody, maybe more. But we are always going to be looking at IgG, immunoglobulin G, and this is the structure of immunoglobulin G. It has two antigen binding sites, or epitope binding sites. It means the same thing. And these are the parts that you should know are randomly generated when the lymphocyte that makes this antibody, when it was young, it randomly scrambled some of its DNA to make randomly generated receptor sites. And that's these two things. And then the other part of it you need to know about is the Fc fragment, this part. This is not randomly generated. This is the same for all IgG antibodies. And the whole point is that lots of cells in the bodies have receptors that detect this. So this has to be constant. It has to be the same for all the antibodies. Okay, well, that's what you need to know about the antibody structure. Antibody function, there's this list. This is the list. Memorize that list. And you can memorize it if you want, just word for word, or you can think about this figure and memorize it in a way that's descriptive, like saying what neutralization means. Because neutralization by itself is not descriptive. It's like neutralizing what? Okay? It's neutralizing protein toxins. Well, that's a useful thing to say. So don't just memorize these words unless you know that they have some context. So these would be better because they give you a little bit of context. Like, these are about toxins. These are about bacteria and viruses. So don't say they restrict bacteria and viruses, because that's meaningless. Say they restrict motility of bacteria and viruses. Okay? So that's antibodies. Make sure you know their structure in detail and their function in detail. So then we move on to the next topic. I'm just going through the study guide here, so I'm looking at it. The next topic is lymphocytes, and several places in these slides, you'll find this slide. It looks like this. It might also look like this, and it's at least one other place. If I scroll through the slides I'm looking for it, it's in here 3 times, and they all say the same thing. The whole point is that this is so important. I want anyone scrolling through the slides to see it and realize it's important. And that's why I give it this nice colorful background sometimes. So, what is a lymphocyte? You need to know all of these things about lymphocytes. Just in general, they are the B cells and the T cells. And if I ask you to tell me about lymphocytes, you absolutely must tell me about their receptors, their special lymphocyte receptors, because that's the only interesting thing about them. Each one of them has a lot of copies of one receptor. And that's what this picture shows, different shapes So this has a bunch of copies of this square receptor, this has a bunch of copies of the circular receptor, this has a bunch of copies of the triangle receptor. So, but each one has multiple copies of those receptors. And those receptors are randomly generated. So the lymphocyte doesn't know what it's supposed to detect, and we don't know what it's supposed to detect. We can't predict it because it's randomly generated. And there is an astronomically huge number of possibilities for what these receptors could detect. So, if I ask you what is a lymphocyte, your answer should sound something like what I just said, because this is what's important about lymphocytes. And I will definitely ask you what a lymphocyte is. So, get ready for that. The receptors, there are two kinds of receptors. There are B cell receptors and T cell receptors. So B cell receptors are what define something as being a B cell, and T cell receptor defines it as being a T cell. You can say both of them are randomly generated, but they're a little bit different because a B cell receptor can bump into something and detect it directly, attach to it directly, whereas a T cell receptor can only detect something if it's presented by another cell in antigen presentation. And that's something we'll get into in more detail in Unit 3. So, cool. So then lymphocyte development. And I'm trying to see, there isn't a single great slide about lymphocyte development, I think. Yeah, there isn't a great slide about lymphocyte development, but you'll recognize from the videos on essentials of adaptive immunity, which you should have watched, something like a diagram like this, where you start with immature lymphocyte at the top, and then there's an arrow. And if you've studied this, pause as I go and predict what I'm going to write, because the next thing I'm going to write is going to be mature, naive lymphocyte. Now you'll notice, if you were studying ahead, if you watched all the videos or didn't use the study guide as your guide, you have seen B cell development and you've seen T cell development. And both of those don't perfectly fit this because they're specific to those things. This is the general development of any lymphocyte, and every lymphocyte follows this pattern with some specialties for B cells and T cells, as we will see. So the next step, so it starts as an immature lymphocyte. The next step is it's a mature but naive lymphocyte. The next step, after it goes through activation, is it is an activated lymphocyte. And this is a very short stage in its life. Also, up here, what am I gonna write? What is it that changes an immature lymphocyte into a mature but naive lymphocyte? It's education. And we will look at education more in Unit 3. So, what happens after a lymphocyte gets activated? The first thing that happens is it goes through clonal expansion. That means it makes as many daughter cells as it possibly can. And then its daughter cells or its granddaughter cells or its great-great-great-great-granddaughter cells, however you want to talk about it, its descendants, differentiate. And they get their final jobs. Some of them will be memory cells, and they will wait for the next chance to fight off the same pathogen, maybe a year later or 5 years later. And some will be effector cells. Most will be effector cells. Effector cells are the cells that do the jobs of the adaptive immune system. So there is, in here somewhere, in the slides somewhere, there's a list of the effector cells. Yeah, here we go. And this is what we call them. These on the left side are the effector cells. And you should know exactly what we call them. So CD8 positive cytotoxic T cell, CD4 positive helper T cell type 1, CD4 positive helper T cell type 2, or plasma cell, also called plasma B cells. And then what they do. So you have to be able to list them, give their specific names, and tell me their jobs. Again, this is an kind of intense thing to memorize, all the little details, but you need them because they will really help you learn how the adaptive immune system works. We can keep coming back to this and unpacking their names and unpacking the details. There's logic to it. There's logic to the job this has, how it works, and the fact that it's CD8 positive. There's logic to these two and their jobs, and the fact that they're CD4 positive. There's logic to the name cytotoxic. There's logic to the name's helper. There's not much logic to the name plasma other than you find them in blood. Right, so that's another big part. We don't spend much time on it because it is just memorize that list, but definitely memorize that list. All right, so lymphocyte development and effector cells. I'm lost. Am I lost? Nope, that's it. So lymphocyte development and effector cells. Looked at receptors, antibody functions, antibodies, and development, and that's a pretty good list of things to know for essentials of adaptive immunity. And if you know all of these things, we can go through them and connect all the dots in Unit 3 so that you'll be able to leave this class with a real appreciation for your adaptive immune system. And it is one of the most amazing things you can learn about a human is how the adaptive immune system works. So I hope you do appreciate that once you learn it, and I will see you at the next video.